The cerebral cortex, like the cerebellar cortex, is organized as folded layers of grey matter overlying the white matter. This section shows the density of cells in the cerebral cortex. The main neuronal type is the pyramidal cell, which comes in various sizes. Differences in the numbers and sizes of neurons allow the cerebral cortex to be subdivided into a series of layers, but these layers cannot be clearly distinguished in this section. These layers extend over the entire cerebrum, but subtle differences in composition allow the cortex from various regions of the cerebrum to be distinguished and numbered. No doubt you'll learn about this in more detail if you pursue a study of neuroscience. Pyramidal cells usually have a prominent apical dendrite which points to the surface. The thinner axons project into the white matter from the deeper ends of these cells, the bases of the pyramids. This section also shows the shape of the neurons in the cortex quite well. Now you see why they're called pyramidal cells. A section of the cerebrum has been stained here with a particular silver technique to demonstrate astrocytes. Silver ions are absorbed onto their microfilaments, staining the entire astrocyte cell, not just its nucleus. Here, in this image, I have found them well stained in the white matter, where conditions were just right. They appear as spidery cells, totally blackened by the silver. When they're suitably oriented, you can trace some of their processes for considerable distances. In particular, you can find them making contact with capillaries. The capillaries are the empty spaces in the white matter. One very important function of the astrocyte is to contribute to the structure of the blood-brain barrier. These processes from nearby astrocytes will completely envelop the basal lamina of this capillary, and similarly, other astrocytes will do the same in most other capillaries in the central nervous system.